Hello everyone. It's terrific to welcome you here today to EPA Victoria's 4th Environmental Science Seminar Series event on the topic of PFAS exposure, bioaccumulation and health effects. And really what we're aiming for today is to talk about what we do know in terms of these group of chemicals, but also what we don't know with special guest speaker Professor Jochen Muller, who I'll talk about later, and it will all be good, Jochen, it's okay, I'll say good things about you. He comes to us from the Faculty of Health and Behavioural Sciences at Queensland Alliance for Environmental Health Sciences, and it's great to have him here to share his considerable knowledge and insights, probably being the first or one of the first to actually look at these chemicals in Australia. So he's got a long history of working with these chemicals and has got a great knowledge of them. Our event today is being held on the traditional lands of the Kulin Nation. I wish to acknowledge them as the traditional owners and pay my respects to elders past, present and future. I'd also like to welcome members of the EPA Governing Board. Graham Ford and Ross Pilling are somewhere in the audience. Welcome today. And a big welcome to those who are joining us via live stream throughout Victoria, Australia and overseas. We have a stunning 535 people joining us today online, so I'm terrified the technology is going to work. So for those out there in live streaming land, welcome. I hope this works for you, and I hope you all get to actually see this presentation. And we also have some media outlets who are using the live stream facility as well, and particularly in the Gippsland region, so hello to that crew as well. A few housekeeping notes. Please switch your phone to silent for the duration of the event, and in case of an emergency the tones will go off, so follow the instructions of on-site staff. So today's series is really to discuss PFAS exposure, bioaccumulation and health effects. And I think all of you are here today and we've got such a big audience because we are finding this material all over the place, all over the country, and because there's an awful lot we don't know about these chemicals and their impacts. There's increasing concern worldwide. We know that this manufactured group of chemicals, there are some 4,700 of them, which is pretty amazing when you think about a group of chemicals. They're resistant to heat, chemical and biological attack, and when they find their way into the environment, they persist. And of course, from a Victorian EPA point of view, we're finding them in soil, surface and groundwaters, and also biota. So we're certainly concerned about them because we know them to be accumulate, and I know Jochen's going to talk about that. And I think the main thing for us is that we actually find them in our homes, and we're finding them in the human population at low concentrations as well. So this is not a chemical that we're finding in particular select locations, although we might be finding higher concentrations of them. And in Victoria we have many sites that are currently affected by point sources of contamination. And then when we start to talk about health effects, there's considerable debate about the health effects of PFAS and at what concentration something might generate a particular health outcome. So it's a legacy issue, it's a current issue, it's got wide spatial extent, and it's something that we need to do something about. So on this note, it's great to actually introduce our guest speaker for today, Professor Jochen Muller. Yeah, I'm going to say something about you first. He grew up in... Sit down again. He grew up in rural Germany near a cable waste incinerator that emitted dioxins, a group of chemically related compounds that are also persistent environmental pollutants. He went on to study the impact of dioxins and other organic pollutants on human health during a master's degree at the University of Hohenheim and a PhD from Griffith University. He became a Fellow of the Queensland Alliance for Environmental Health Sciences at the University of Queensland, which used to be called NTOX, where he established a research group that established and focused on exposure to chemical pollutants, including novel approaches for actually assessing exposure. His team commenced work on PFAS in 2004, so he's got a good history with this particular group, and he's demonstrated exposure to various PFAS over the last decade. As part of his research, he's led numerous national and international studies, such as Australia's National Dioxin Program, the Brominated Flame Retardant Program, and a major study to investigate bushfire emissions, and the results of which are included in the United Nations Environment Programme Toolkit. So it's my great pleasure to introduce Professor Jochen Muller here today, so please join me and welcome him to this talk.
Thanks so much, Andrea. Thanks very much. This is a bit intimidating, I have to say. I don't think I have ever really talked in front of 500 people online. Okay. Now, firstly, research is a lot about... Firstly, let me also acknowledge the traditional owners of the land, and it's really great to be here. And thanks for the WikEPA for inviting me. And I like to start off with thanking a couple of people that are on my slides. I don't know whether this is going to work. No, maybe not. Oops. So first of all, there is no good research without good students. And I like to acknowledge a couple of my students, Lisa Toms, Jennifer Bräunig, and also Jack and Christy Thompson. All of them have worked with me on PFAS. And a few great colleagues, Lisa Aylward and Antonia Calafrat from the US CDC, and I will talk a little bit about their work. In my title, I try to cover a broad spectrum of PFAS things, what we know and what we don't know. And I thought the audience would know probably a lot about PFAS already. So I tried to bring a couple of curiosities in. For example, I take you along the path of why do they bioaccumulate? Or at the end I give you some personal notes, and I'd like to say right at the start that I have to take full ownership, and I may not, there may be quite many people which don't share my final conclusions, so it's my personal views there that I express at the end. And they actually come from something that Andrea mentioned right at the start, which is that I came from a contaminated site in a small town in South Germany where there was a cable waste incinerator incident, and we all thought we're going to die of cancer very soon because there was high levels of dioxins around us, and it scared us really all in the mid-80s, and this is why I actually studied what I studied. This is how I came to study environmental toxicology. And now I think the scaring was worse for us than actually the toxic effects of the dioxin or the exposure. And that leads actually to some of the conclusion I come later. But let me go through my talk. So I have an outline. I will give you a very brief history of PFAS. And each of these subtitles, I will end up with a slide saying what we know and what we don't know. I will try to summarize what we actually don't know. So quick about PFAS history, what they are and how we analyze them, then something about exposure and pathways and routes. Then I take you along the path of why do they actually bioaccumulate. I think this is probably what is least understood. Why are humans so different? And we are actually different when it comes to PFAS. I tell you some good news about serum data in Australia, at least for the general population. Then I take you a small path on the health and toxicity, and then I turn this around with some personal thoughts. And I came out from an overseas trip last night, so if I snooze off in the middle, I have something to keep me awake to get back. This is a simple PFAS timeline, and this is actually stolen from a guy called Ian Ross who works from Arcadis. Some of you may know him. He's a big promoter of something called the TOP assay. And he put this together. What I'd like to say here is they were invented, they are old actually. They are older than even DDT, or around the time of where DDT was developed, the PFAS apparently it was almost an accident. And they were then in the 50s developed into products, and in the late 60s they were actually following a ship accident, a ship fire. They were turned into a firefighting foam. That's my understanding. And around 2000, The first time I heard about it was around 2000 from a guy called John Giese and Cannon. They gave a talk at the dioxin conference, and I thought, what are they talking about? These chemicals don't have chlorine. We usually only care about chemicals that are chlorinated. And they are aliphatic, so I was really, I never thought I'd get into this. I remember then Jack Holland thought they were quite curious. It was the next person I talked to about this. But anyway, around 2000, environmental scientists, environmental toxicologists became aware of them, and then sort of a big slow frenzy started, and then I think in Australia the thesis hit the fan about 8 or so years ago, slowly, bit by bit. Now, I also put on the bottom something about the two ways of making PFAS in general. There's an electrochemical fluorination, which produces 70% linear, and I come what is linear and branch, and 30% branch, and there is a fluorotelomerization, which uses something like fluoroethylenes or fluoroalkenes to make them, and they mainly produce short chain, even numbered chemicals. Okay. And then we have two groups of these chemicals that we generally, when we call PFAS, we say poly and perfluorinated chemicals. I know this is probably most old stuff for you guys, but I just like to, when I say PFAS, I mean both. And when I talk about PFAAs, then I mean the right ones. It's the polyfluorinated alkyl acids. That's where PFOS and PFOA is part of. And they are, carefully said, very difficult to destruct. You need a lot of energy to essentially break them down. And then on your left-hand side, there is the polyfluorinated substances, which have these carbon stuck in here. Carbon with hydrogens, and that makes them weaker, and that's where usually they break to bits and may be the precursors for the PFAAs, the ones on the right side. And as Andrea said, there are 4,738 or something like this. Who counted them? I don't even know whether they include the branched and the linear separately or not. And they come in all shapes and forms, and the perfluoroalkyl acids are very persistent, and the poly have a huge variety in their properties and so on. And when they get bigger, if we go from a C4 to a C12 or something, then they become less polar, less water soluble. And when they have a sulfur groups, the perfluoroalkylic acids, when they have a sulfur groups, they are more non-polar than if they have a carboxylic acid groups, which has an effect on how they behave or how long they retained in the soil on the way to, for example, the groundwater. So shorter, travel fast, carboxylic acid travel faster than sulfonic acid. And then there are precursors to them. They have all kinds of moieties, and they can be anionic, zwitterionic, or cationic. Okay, and they can fall apart, or they can be transformed biologically or chemically to perfluoroalkyl acids. And chemical transformation does not necessarily predict biological transformation. So that makes it all confusing. We use this assay, and I come back to this, which does a chemical transformation, but it doesn't predict actually what's going on in biology. Okay. Just to make it confusing here, a list of chemicals from Jennifer Field and Chris Higgins' group from Bas and Hansen. And each of these chemicals, there are numbers there in this table. I know you can't read it. It's not for you to read it. You can read the paper. But there are numbers there, and these numbers says how many different carbons can be in this. So every of these chemicals can have multiple forms with multiple carbons. On the right side of this figure is when you look at wastewater treatment plants in Australia, what goes into the influent of wastewater treatment plants and what comes out of the effluent of wastewater treatment plants, then you see that, for example, for PFOA, there's almost 10 times as much PFOA coming out of wastewater treatment plants than are going into wastewater treatment plants. Now, two explanations to this. Either somebody chucks in PFOA in the wastewater treatment plant, for example, by doing something funny, or there is something going in that we don't see that converts to PFOA. Now, we saw the same for PFOS a couple of years ago. We have redone this work in 2016 on census at 100 or so, or 70 wastewater treatment plants, and we couldn't see PFOS anymore being more coming out than in. Now, we are not sure whether our result earlier was not right, but for PFOA we see the same. For the sulfonic acid, it's not published yet, but for the sulfonic acid we just see roughly, give or take, what goes in comes out. Now, there is something also going into the biosolids, and 70% of all I say is made up on the spot. No, no. But there is something also going in the biosolids. Let's say 10%, 5 to 10% of the PFOS. Most of it is coming out. We're not quite sure whether there is much PFOS produced in the moment in the wastewater treatment plants. Here's an OECD pamphlet that came out last year, and it essentially says that of the 4,730 PFAS that are identified, about 90% may be precursors. Now we cover, here it says 14 with standard EPA method. Depends which lab you work with now. You can do 29 or 37, or I think we are now up to 45, or something like this, different PFAS that you can measure. But we are a long way away from the 4,730 recognized ones, and there are other non-recognized ones. This is a study we did trying to measure PFAS mobility around the firefighting training ground using a suspect screening method. So it's a method that doesn't use just standards, but it uses also other methods. It uses a mass. We're looking for masses that have fluorines in it that are organofluorine chemicals. And we actually could sort of identify these chemicals. They have all acronyms there. And the light blue, the dark blue ones are the chemicals that we have standards for. The light blue chemicals are chemicals that we don't have standard for. And down here we look at how deep these chemicals are traveling, which tells us something about their mobility. All right? So this is 0 to 2 meters around the firefighting training ground. And you can see that depends on the chemical, some travel fast, some get stuck in the top layer. And we think this is because of these different properties. Okay? Now we have to say, the analytical method that we used for this, the extraction method of soils, is based entirely on extracting PFOS. Now if a chemical is, for example, a Zwitter iron, we may not actually be good in extracting it, and we can't put a standard for that chemical in because we don't know it. So our analytical method is rather dodgy, because we rely on an extraction method that is essentially the extraction method of PFOS. By the way, you can interrupt me or shake your head if you don't know whether, you know, not those people on the internet. Okay. All right. And the bad news here is they are a bugger to remediate. They are really, I mean, actually what remediation does is we are moving them forward and backwards and trying to make them smaller in the process. So when they are in water, when I go to Europe, I put my Aussie accent on. When I'm in Australia, I put my Bok accent on. All right. So when they are in water, we take them out of the water with the sorbent, separate them, but then we have them in the sorbent. If you guys, at some of these remediation sites, help? There was only one button to click. That's how I keep you awake. Always. All right. So when they are in the water, we take them out of the water, put them in the sorbent, and on these remediation sites where they remediate water, they build up sorbent. And then when we have them in soils, our idea is we put them then in water, where we then put them out of the water back on a sorbent. Now this is really true. These are the methods we have. The problem is that actually in the environment, the concentrations are still very low. I remember when I was an undergraduate student, there was a newspaper article. 9 nanograms per kilogram dioxin found in the soil. And my mum said, "Oh, nanograms, that must be a lot." The reality is, these chemicals are in the environment at concentration that is essentially point bugger all. And it makes it not worth to do something with this environmental phase directly. So what we have to do, we have to get them from these nanograms over many orders of magnitudes, enriching them to ideally milligrams, so they start foaming again, and then we can maybe make it worth to put the energy into it to destroy them. That's really the goal here, because there is no method. And by the way, there are some people who hope that we can find bioremediation that bugs eat them up. Now, if you would be a bug and go through the soil and you rely on PFAS for your food, you must be bloody desperate. And that's the reality. It's quite tricky, I think. There are so many other energy sources in that soil that I think to rely on such a solid bond CF, I'm starving for CF. No, this is really unlikely. Anyway, and last but not least, they are an analytical nightmare. So I'm taking you a bit through analysis briefly. Now, who has done PFAS analysis in the room? Okay, good. One. All right. No, that's great. So that's good, because then it's worth to explaining. So we have 2 types of approaches to analyzing. One of these approaches, or the main approach that most of you, if you have ever commissioned somebody to analyze PFAS, has probably done a target analysis. You say, I want the standard EPA 20 or so. These are well-defined target compounds. We use LC triple quad mass spectrometer. We use something called isotope dilution method. We actually put PFAS, these 20, into the soup, into the soil, into the water, in whatever the sample is. We put them in, but they have carbons that are different mass, and we can then later say, we put 100 in, and we found a peak that is double as big than the peak that we put in, and we know what we put in is 100, then that other peak is 200. That's how easy it is. It's actually all relative to what we put in. You can even spill half of your sample by accident. And you can still say it's double because you assume that what you spilled as much of what you put in than what and you get a recovery. And the US EPA actually accepts ridiculously 20% recoveries. That means you spilled actually 80% somewhere, you lost it, and you still accepted them. Now that's what we call target isotope dilution method. It's a really good method. There shouldn't be much doubt about it. People do make errors, I know, and there are inquiries about these errors, but normally this method works. Okay? And then there is a second method. It's the LC-QTOF method. That's sort of, it can do target analysis, but actually the QTOF, the first instrument, this LC triple quad, it only looks for the masses that you tell him to look. And that's only, if you look for 20 chemicals, it's actually 40 masses, because we look every chemical with 2. And also your isotope dilution. But the right one, the right method, it tries to look for all masses. It sees thousands of chemicals, and I show you quickly a slide. It tries to, you can use it to do accurate target quantification, which means you can still use isotope dilution method, but it's much more tedious. It takes more training of your staff. It's more tedious to actually do the quantification. QTOFs in the moment, the best ones are probably 3 to 5 times less sensitive for the common PFAS, the PFOS and PFOA, than the normal. Then the most important thing is you can retrospectively interrogate your mass spectra. Sorry that I'm boring you with this, but you can look back in time in your samples because everything is archived, and you can look, well, now I have a new PFAS that I think about. I can look back in time for this new PFAS in my archived data, okay? So it's the new kid on the block. All the big guns in the field, Jennifer Field, Chris Higgins, ourselves, just kidding, they are moving to do the non-target, at least to some, and I come back to this. And then there is, this is how a non-target, this is actually a blood sample looking for perfluorinated chemicals in blood samples of firefighters. This is how it looks. These are all the peaks that we find in a certain area, and it's like finding really a needle in the haystack. But then we refined our methods, and we found needles. We found new chemicals that are associated with, that are perfluorinated chemicals that are in firefighters that nobody knew before. Chlorinated PFAS, actually. But anyway, and then there is the other thing, there is this top assay. Who's heard of the top assay? Let's ask the other way around. Who hasn't heard of the top assay? Okay. So the top assay, 4,700 chemicals, PFAS. Now we would actually like to close the mass balance, or we would like to know what will be our future legacy problem for PFOS and PFOA and so on. So it's a method to hit the broth with a chemical that converts it for us really fast and tries to allow us to predict what may happen. Now, it doesn't predict what biology does to the chemical, to the precursors, but it does predict reasonably well probably what the chemical conversion is. It's called the top assay, developed by Houts and Sedlak. And people use the top assay, at least to some extent, using, typically it's used with the target analysis, and that's where it predicts how much PFOS is formed. So you do a sample, you analyze it for PFOS, let's say, and then you throw in you do your top assay, and then afterwards you analyze the same sample again, and you look before and after, and you will find out whether you had formed new perfluoroalkyl acids from chemicals that you actually don't know. Now, the great news, I think, if we want to do the top assay, we want to actually know what were the precursors that cause, because that's what we want to regulate. That's what we want to take out of the system. So for this, you actually have to do the top assay with the QTOF, the non-target. You have to see what has disappeared since we looked before and identify this. Very quickly, one slide. Total oxidizable precursor assay, my take of it. It was originally developed for aqueous matrices. I think some ambitious Queensland regulators in the room, not just Tony, I didn't mention names. Some regulators have broadened that application to other matrices that maybe actually that causes a bit of headache because it's quite tricky to control this. It does not close the mass balance, and I think it's really a tool for us to understand it better, to regulate based on the top assay. Personal opinion, maybe that's a little bit early. I think for aquatic matrices, great. I think for some other matrices it may be tricky. So my first slide, this was a bigger sort of piece. I think we need inventories for what PFAS we are actually releasing, and we should try to get a hold of past inventories. We have to do more about transformation and what chemicals transform to what and how fast. I think we need routine sampling methods as much as routine analytical methods. And I think a lot will happen in this space. And we need to understand something about the mass balance. What is actually out there? What do we bother? Do we bother about everything? It takes a lot of effort. Second part. Okay. So exposure sources and exposure types and routes. So for this, I tried to make a few new slides. I hope they work actually. So when it comes to exposure sources and exposure types, and I really call it both, we have first of all these occupational exposures. And we mentioned particularly firefighting foams. We all know that has been a source of exposure, for example, to firefighters. And then the second thing, I think that's a clear one, is use of consumer products. Use of consumer products. It's actually not really well understood. I come back to this when I show you our serum data, because I think that has played a big part of why we see the levels decreasing. And then we have these exposure routes that are indirectly, and in exposed communities, we're really dealing with the use of firefighting foams that led to contaminated drinking water and contaminated food and contaminated dust. Whereas the consumer products are stayed the same. We know that PFAS is in the water. Actually, we did this study close to 10 years ago as Jack Thompson's PhD. And we analyzed drinking water from around the country. By now there should be much more data. Actually, I don't think anybody has collated them, but I think there are efforts to collate them. But you can have now a much more comprehensive data set from Australia where PFAS has been in the drinking water. This was an old study where we found that on average the PFOS concentration was about 1.6 nanogram and the PFOA concentration about a nanogram per liter. And we found that almost all sites had one of the PFAS that we could measure in those dyes. We did go very low. We did analyze in our report in the sub 0.5 nanogram per liter. And at the time we did the backwards mass balance model where we estimated from what we find in people back what that probably means in intake. And we estimated that drinking water at those levels, even at the highest contaminated sites, contributes only to about 10% of the total PFAS burden. PFAS in food, they can be found in food. Fessans did a study. They actually found it only in very few foods. I don't know whether they did another study. Probably some of you in the room know this much better. But there are overseas studies. Arnie Schechter in Texas did a big study on PFAS in food. He collated a number of food, found the highest level of some of them in olive oil. There are other studies in Europe. I remember there was a bit of kerfuffle maybe almost 10 years ago about PFAS in potatoes. But the evidence that food is an important exposure pathway comes in part from questionnaires and people where, for example, it's shown that people who eat more fish, there's a correlation between higher fish consumption and PFAS in blood. I think food is more difficult to assess because of the variety of food we are dealing with. I think food was also a problem because do we measure the food after it's been wrapped up and unwrapped, or do we measure the food fresh? We've done a big study with New South Wales food. What I can tell you is, in our study, when you cook fish, the normal way, you're neither adding PFAS unless you use a really silly pan, and you're not destroying PFAS. You're transferring some of the PFAS in the cooking oil or in the water, but you're not actually, but it's very small. The PFAS mass balance in the cooking, this is a study of Matt Taylor, I have to mention. There is no destruction. There's just a paper coming out of Turkey where they cooked fish for 20 minutes. Now, Matt Taylor said if you cook fish for 20 minutes, you don't eat it anymore. But maybe there is a particular dish in Turkey. But anyway, they said that the PFAS level in the fish decreased by almost half. That's just been published. We couldn't see any of this in fish that we would have loved to eat. Okay. In terms of occupational exposure, and I use these symbols, I used these introduced before 5 symbols: occupational, dust, food, consumer products, and water. And so I think in occupationally exposed people, we think it's something to do with occupational exposure. We actually don't know how certain firefighters were exposed. We did a study on firefighters with Air Services, it's published, where we showed that the only thing that we could link to high serum levels of PFOS was that the firefighters have worked before 2004 at Air Services. All those that were later than 2004 didn't have higher levels. And we think this is because before 2004 they used light water, which was a PFOS-based foam. When you come to places that have a contamination source, we see something really different. Actually, when you come to some Australian contaminated places, you find this redistribution of PFAS. And you see this in consultancy reports, and we wrote one paper about it. But when you actually go there, you see that they may have stopped the use of groundwater for drinking water, but the groundwater is still used for sprinkling the garden and for doing all these other things. And I remember the first time I came to this one town, there were sprinklers going, it's very arid, sprinklers going, the dog was playing in the sprinkler and the kids were catching the dog under the sprinkler. And I thought, gee, this is really difficult, how distribution of chemicals and exposure pathways work. And we really don't understand this. What we understand is that the PFAS in these communities, when you look at the sites, is elevated essentially throughout their products. Now, I don't have actually control samples, but the control sample here should be essentially near zero. And when you look at, particular in this case, some of the beef and some of the other samples, it's really quite astonishing how high the concentrations were. And this, I think, Roger Drew here in the audience, he actually does a study, how long does it take to clear the PFAS out of these cattle. It's really interesting that a cattle clears the PFAS much, much faster than a human being. So that's, although the body mass is much, much greater, which brings me back later to the bioaccumulation. So, if we talk in exposure in communities with with contaminated groundwater, I think water and food are the big drivers. Dust, maybe, food-mouth contact. So this is sort of a conceptual picture. When it comes to the general population, we think it is food and maybe consumer products. There is a study from Winkins, Kirsten Winkins from Ian Cousin's group, who suggested that actually consumer products contribute about, well, actually precursors contribute about 8 to 10%. And when it comes to babies, although PFAS are not very lipid soluble, they are found in breast milk, and actually breastfeeding does make a contribution. I didn't think this really first, but it's the evidence, the latest papers suggest that there is some contribution from breast milk. So, what we don't know, we really struggle understanding exposure routes. And that makes it actually hard to do the interventions, to make sure that we are cleaning our act up. There has been a recent paper that showed that when you stop, when you give advice, people do go down faster. What's the paper? I have to think of the author, the main author. He's one of the C8 studies from the epidemiology group. Comes back. Anyway, so what we don't know is current advice in communities and occupational cohorts actually working. And this is why in the moment we have done only usually one cross-sectional time points for these communities. So for most people we only have one point. And I think we need other points to really see this is working. We really don't know the role of consumer products, and we don't know the role of precursors. So, half time. A bit more than half time, but to keep you awake, why can't you use beef stew as your password? It's not Stroganoff, yes. That's right. Great response. Dad joke. Tony told a dad joke in the break, but I can't get it back, about tortoises. Okay. All right. Are you with me still? Next thing is a little bit dense, but possibly the most interesting one. Okay. I'm doing a bit of pharmacokinetics. Have you ever wondered why PFAS bioaccumulate? Nah. Who cares? Why do some bioaccumulate? Okay, so in pharmacokinetics they talk about this ADME, adsorption, distribution, metabolism, and excretion. And I take you briefly through this in humans, okay, as much as I can. So we know PFAS, when it's consumed, is fairly well absorbed, and there is a decrease in absorption with chain length. Now the absorption through skin is low, depending on the ionization state. It can be high if not ionized, but it's usually ionized, so it's really not usually considered a problem. And these chemicals also absorb in plants and worms. And just one little slide from our own work, this is a PFAS contaminated soil. When we reduce absorption by mixing the soil with something that absorbs the chemical in the soil, or that essentially reduces leaching, it's called a chemical called Rembind. When we do that, the concentration in the leachate decreases very much by orders of magnitude, by 2 to 3. No, oh, sorry. This is log scale. So if you go one level down, it becomes tenfold down. So it's a big decrease. So 99.9% of some of these decrease for PFAS for the more sorbing chemicals. When you do that, what happens when you put this, when you put worms in the soil? Then you also see, then when you put when you inhibit the bioavailability, then you get also much less uptake in the worms. So this means that absorption is bioavailability is really directly related to the availability of the chemical or the desorption of the chemical. And the same in grass. So very short story, if you limit, if you bind the chemical in the soil, it becomes less mobile and it becomes less available into grass and it becomes less available into worms. And it's a matter of what's your chain length of your chemical. So here I plot from C4 to C12 carboxylic acids, and here is the sulfonic acid. I know I'm challenging maybe a little bit. Now, this was absorption. I'm now coming to the distribution and storage. So PFAS, how do they distribute and how do they store in our bodies? They are non-covalent bound. That means they are not forming a chemical bond, a chemical binding with the plasma and the lipoproteins in the plasma. And there is this balance between what is bound and what is free. And a very small portion of the PFAS that is in your blood is actually in the free form. So where you find the PFAS in your body, it's in protein-rich tissue, such as your liver, your kidney, and so on. And we toxicologists think that the bound PFAS is not active, but actually acts as a reservoir. And then there are these organic acid transporters, which I come back to. And on the right side, there is this binding constant, which is in a paper from Ng and Hungerbühl in Switzerland, where they show that there is a certain chain length where the chemicals bind most tightly. And these are about 8 to 12. So these chemicals bind more to your protein. That means they are less excretable, they are more caught in your body. So that's about the distribution and storage in your body. When it comes to metabolism of the chemical, the perfluorinated chemicals, we think, do not degrade at all. There is a chance that the polyfluorinated chemicals actually form, the perfluorinated chemicals form from the polyfluorinated chemicals. This is again what Winkins et al. Suggested. About 10% of what's in your body comes from polyfluorinated chemicals. And it's also put in a model from Mittal and again the Swiss group. They put this even in a model, this transformation. And Chris Higgins in the US is in the moment doing a study particular on this transformation, because essentially we have banned PFOS and PFOA. So now we're dealing only with the release of potential precursors. Now, if we get exposed to these precursors, will this result in future transformation in our body to stable PFAS? Now, the big trick, and I think this is what really I didn't appreciate until a year or 2 or 3 ago, it's about the elimination. This is the trick. This is where humans seem to be really different. So people write very easily about this. PFAS are then reabsorbed in the kidneys. So these fatty acid transporters that when you excrete, so essentially the PFAS gets transferred into the urine, but then there are these really very efficient transporters that transport the PFAS back. And they're not only in your urine, they are also actually in your guts or in your intestine walls. And there is multiple evidence in this. For example, there is a study where they gave somebody, or that they looked at cholesterol-lowering drugs, where they showed that these cholesterol-lowering drugs actually, which work on the fatty acid transporters, I think, in the gut wall, they also inhibit the retransport of PFAS, and you have more excretion. So essentially, people are thinking now of the idea whether there are medications that we could give to prevent the reabsorption and speed up the excretion. Okay, now we are falling all asleep. But essentially, what I'm trying to say, bioaccumulation is complex, and where humans are really different is probably on this reabsorption bit, where, because I think blood of animals is very similar in terms of protein composition, so it's this, we are not eliminating it as efficiently. Yes, I think I skipped this, I think I summarized this, but there's, so, and I said all this. There's also something about this on sex differences, particularly in rats. Now, are we looking for the right PFAS? Now, there are other PFAS that are actually accumulating. This is, for example, these PFAS that we found in firefighters that are not the conventional PFAS. There is a chlorinated PFAS. There is a ether-based perfluoroalkyl substance. And there's recently a paper from China which showed bioaccumulation of a whole range of other PFAS. So there's not only these PFOA that are bioaccumulating. So you will hear probably more from the space of whether these other chemicals are. What we don't know about bioaccumulation. The difference in accumulation in different people, is that to do with our differences in ability to excrete PFAS? So there are people that have extremely or very low PFAS levels, it seems. Is this because they are less exposed, or is this because they are more excreting the chemical? And is it feasible to speed up this elimination? Can we use drugs? We know actually blood donors have lower PFAS levels. Hemochromatosis patients, we did studies on hemochromatosis patients, have lower PFAS levels. And why do different species show such great differences? And then we probably in the future will hear a lot about the role of precursors. Okay, the easiest part of my talk is actually jumping from this bioaccumulation to the good news, the serum study. So we've been doing since 2002 a study initially funded by Sarah Broomhall's predecessor, Chris Mobbs and colleagues. And what they wanted in 2002, they asked us to look at what is the average concentration of dioxins in blood in Australia. And for this study, initially we collected 10,000 blood samples from surplus pathology samples, and we pooled them according to age and postcode and gender, and we created 100 pools. But in short, it was such a successful study that we decided we do this every 2 years. And so here on the right, you see this timeline where every 2 years, and the S stands for a particular study, every 2 years we collect nowadays 2,400 blood samples, cross-sectionally leftover pathology samples, and we look at a whole range of chemicals in those bloods. And initially, Antonia Calafat analyzed them all for us for free because she thought this was really quite curious. Yeah. And this is a set of data just for PFAS. So what you see here, the red are the 2002-2003 samples that were collected then, and then the green, and so on, you can see. And at the bottom you see the 2016-2017. Okay, I should have explained this graph a bit better. On the x-axis you have age. Every dot is 100 sample on this figure, except some of these dots on the left here, these green dots, because we didn't have that many children. And so what you see essentially is the levels are decreasing. So you can see that, for example, for PFOS, the PFOS levels in the Australian general population, now all the older people are put into one big box for each of these years. And you can see that the PFOS concentration in the Australian population has decreased by 85% or so since 2002. That's a brilliant news. So this suggests that exposure has decreased by a substantial amount. Interesting, hexasulfonate has decreased much less. And it's funny with this non-carboxylic acid, it actually had a peak in 2008. So we don't actually understand yet what led to this different time pattern. And we use this data with Ian Cousins and his team, who are modelers, and Matt McLeod. We have used this to backwards estimate from the sum of all these data to backwards estimate and enhance a similar data. When was peak exposure of PFOS in Australia? And this suggests that the best fit model suggests that peak exposure for PFOS was around 95. Early 90s, 95. So probably somebody knew maybe we should slowly replace what we do with change in consumer products. And before us environmental scientists actually became aware of the PFOS. Now this is a model, but there is other evidence to this. So all we can see since 2002, the levels of PFOS in the population is decreasing relatively rapidly. By the way, here I drew a line in, and this suggests that our current intake, give or take, from this model is about 1 nanogram per kilogram body weight per day, roughly. And we use this data to estimate what is a high level in the population. So we assumed distributions, and for these distributions we used the National Health, Environment, Nutrition, and the National Health and Environment, Health and Nutrition Environment Study, NHANES data, and they have individual data. They do with thousands of people every couple of years. And from this we get a distribution or a skewedness of the distribution, and we done it also with the Belgian group. We looked at individual general population data and looked at the distribution and calculate what is the ratio of the 95th percentile to the mean. And we just redid this for the 2017 data as well, and just submitted this. But this is the old data. And then we use the statistics from the data, to estimate whether people are higher or not, whether people are normal. So for example, here we compared the firefighters that we looked at in air services, whether they are high. And then we looked further, what differentiates them. And here, I don't know, can you see this mouse? Okay, all right. You can see that if they worked less than 10 years in the job, they were normal. Only if they worked more than 10 years in the job. And so we used the population data to do this. By the way, we also looked at health indicators in these firefighters, such as cholesterol, which is associated with PFOS levels. We couldn't see anything in these firefighters, but there's really relatively few data. So what we know actually with the serum data, levels are decreasing. We don't know why they decreased from the mid-90s. We don't know it for individual people, how they decreased. We are hoping that we can estimate in future peak serum levels, and I think we need to look broader about new perfluorinated chemicals. Sort of summarizing the serum. I'm slowly winding down. So now I'm at the health impacts. Now this is the area where I feel least comfortable to talk about. because and that's the crux, of course. What are the health problems? What is the evidence? What do we know about it? There are other people here in the room which are probably better qualified to talk about this. But in brief, we measure health effects using epidemiology and toxicology. And epidemiology tells us a whole lot of things. The C8 study is probably the biggest study, more than 60,000 people, many elevated, and they found evidence for increased serum lipids, uric acid, altered thyroid, ulcerative colitis. They found 2 cancers, bladder cancer and kidney cancer, I think. And Phil Grosjean is talking a lot about immunotoxicology and immunization. Other studies don't find the same thing, but they are not as powerful, certainly, as the C8 study. And there are maybe many confounders. Like to remind you, correlation is not causality. There is some reason why if you have higher cholesterol, you could also have higher binding capacity in your blood for PFAS, so there could be not causal, and so on. And then there is evidence from the toxicology in animals. I mean, if you look at the molecule, you see that this looks like a fatty acid in broader sense, and in affects fatty acid metabolism. So there is a weight of evidence type thing. So there's lots of evidence. I think that the trouble is there is no PFAS-specific disease. And once there is no PFAS-specific disease, it becomes extremely difficult to find links between exposure. And I think this is the real challenge that we have to explain to communities, that our tools to actually relate chemical exposure to effects are pretty bad. They're enormously bad. And it makes it so difficult. And just because we don't know, and the interpretation of this information that we don't know is so difficult, because there are some of us that interpret this as, oi, that's bad, and others say, whoa, there's no evidence. And this controversy makes it so hard. So I think this is what we have to learn to communicate. So the current estimated background exposure I mentioned before is about 1 nanogram, which is well below the health-based guidance value of that Phesan's put out of 20 nanograms per kilogram per day. But the proposed new health-based guideline value for PFAS from Phesan's is 30 nanograms per week. I don't know, is this now out? I think it's out. It's out as a draft, sorry. Okay, so this is very close to current intake. And the Germans have published a reference value. And in this new paper which we just submitted, we find that the German reference value for which they say there may be, or there may be health effects, is 5 nanogram per milliliter, which is roughly our Australian current PFAS average. And it's well below the 95th percentile in our Australian population. Now that's a little bit, we really struggled how to put this actually in this paper, how we discuss this. We actually predict that in about 10 years, we will be with our 95th percentile below that. What we don't know, health effects. Is health affected? and the causality and mechanism. I looked at ATSDR site last night, and in some of the advice to the people, they say, we know that PFAS causes health effects. We have said in the past, we don't know whether PFAS causes health effects. I don't know. It's not something for me to answer. But if we think PFAS causes health effects, then we should seriously consider whether we need interventions. And we know that we have intervention strategies that are actually easy to implement, at least in the first cut. We can go blood donating. If we donate blood, we reduce our PFAS blood levels much faster than if we don't donate blood. And everybody considers donating blood as something that is non-invasive or not. But I don't propose this. I just say if we think it causes health effects, then the consequence would be that we should think about, should we do something about it? So far, no government in the world has, as far as I know, advocated to do something when you have high blood levels. So if we don't do something, we must think. And then we need to learn how to communicate uncertainties to people. How do we interpret uncertainties? I think this is the trickiest. And here comes my wrap-up. Uncertainty actually causes stress. And PFAS contamination in these communities is objectively a stressful event. And there are all these effects in these communities. They are worried about their health, but they are also worried about financial loss and the future of their community. And they are frustrated, and even frustration causes stress, and stress is unhealthy. And I think I recently listened to a podcast about depression, and there was a big discussion about lack of control is associated with depression. And I think what in these communities is happening is actually there is a lack of control to some degree. Firstly, they're not responsible for the contamination, and then they can't do anything about it. It's so out of control. So, and all this causes emotional responses and physiological. I don't have to tell you, it's a very obvious one. I mean, I'm dwelling now in social science, which is no area. But my suspicion is that the uncertainty stress causes a bigger effect on health, at least a more defined effect on health than the PFAS health. But that's a wild suspicion, actually. Okay, I take this back. Sorry, I take this back. I think what I wanted to say is we better understand the uncertainty stress than the direct PFAS toxicity on health. I think that's what it is. I am not saying that PFAS doesn't cause health effects. It's just really difficult to measure this. So this is, I think, my last slide. What we can do is we can try to reduce stress and help to gain control. I don't know how, but I think this is what we have to try, and support these communities. I think we also want to do everything to reduce exposure. We want to get this whole thing under control. We want to educate about risks. I think education reduces stress. Understanding that uncertainties by themselves is not a risk. I come from university. I like the independence. If we're worried, maybe intervention, maybe even intervention reduce. I don't know. Look, this research relied on so many people. I put this last night together on an airplane from Europe. I probably forgot a lot of people in this. I don't know. But a lot of people have helped me better understand PFAS and helped me to know that I have no idea about it, actually. So thank you very much.
So we've got some time for some questions from the audience. So,
have the manufacturers altered the way that they sell these products into the marketplace? Say like in 1995, seeing that correlation in the dip in the blood serum levels?
I don't know the answer to that, sorry. I suspect, I mean, even the way the PFAS were produced has changed, and I'm sure since the awareness is there, it's a much tighter system. I don't know, but I don't know really the answer to your question.
Hi, I'm Michael Abramson from Monash. I really enjoyed that talk. Thank you so much. There was a time, of course, when doctors recommended bloodletting for just about everything, so many of us would probably be reluctant to go back to that, but it is certainly an interesting suggestion. I just wanted to ask about the human biomonitoring program. Given that they're pooled pathology specimens, many of these people may have had some illness. Was there a measure, for example, to exclude those with known renal or liver disease?
No. Short answer. Actually, in 2002, before we started using pathology samples, we did exactly that, but for dioxins. We collected 400 samples, 200 or 400, I forgot. They were from pathology random samples, and then we had the same amount, postcode, age, and gender matched, that were insurance samples that were for changing insurance. So we called them healthy versus sick. And we did this for dioxins and I think PCBs, and we found no difference between the two groups. And we recently did the same thing again because we couldn't find our 2002 samples anymore. We did the same thing again recently, and we couldn't find any difference for PFOS and PFOA. We did find some differences for hexasulfonate, but we are going to redo it to check that, whether that is sticking. Okay. But a good question, thanks. We get asked that question, by the way, how do you know you can use pathology samples whenever we publish any of these leftover pathology samples?
Thank you for a very interesting and enlightening and honest talk. I just wanted to ask a question. You know I'm a biologist. A couple of questions. One is does it go more in the blood cells than the serum? Are PFAS bound more in the blood cells than in the blood serum because of the proteins? And secondly, you know, there's a controversy about biomagnification of food chains. Joachim, from your understanding of the chemical itself, what side are you on? Is there biomagnification, like in the chlorinated chemicals, or would you say that it's just eliminated and therefore it doesn't sort of magnify up food chains?
I mean, we measure it in serum, but I assume that has the protein, lipoprotein in it, the way we get it. I don't actually know the answer really to your first question. I think the biomagnification is much less. I mean, biomagnification is something to do with KOW typically, or octanol air water partition coefficient. Of course, this does not work for these chemicals. So I think it's here much more a question of specific species. It's actually quite weird what chemicals have more PFAS than other. For example, shrimp in Williamtown, where I think crustaceans were higher, and I think Matt Taylor and Carl Bolt suggest this is to do with benthic. But I don't know. I don't actually know the answer to both of your questions very well. Has anybody got an easy question? Roger, an easy question. Your question always easy.
do I have to press anything on this? No. In relation to the first question, there's been several studies looking at the distribution of PFASs within blood, and they do not partition into red blood cells. And so that's one of the reasons why people concentrate on measuring serum or plasma. I think you're right, Jochen, in that the key between the distribution and accumulation in species relates to the transport proteins of the organic acid transport proteins, OAT and OAT-P, but also in the fatty acid binding proteins. The binding to serum protein, most of it occurs with albumin, but floating around in the serum are transport proteins which are actually used by cells to accumulate or take up materials. And I think that that's where or how primarily these things are getting inside cells.
Any other questions?
Great, thanks very much, Jochen. Tony Merritt from New South Wales Health. So I could have lots of questions, but you just pick one. You were talking about the health impacts from the C8 study, and my understanding there that was PFOA, and in Australia we're largely dealing with PFOS. So I just wondered if you'd make a distinction between those two from all that you've had experience with so far in terms of kind of likelihood of health impacts.
I don't think there is enough real data, but again, this is beyond me. You shouldn't ask me questions that I haven't covered in my talk. It's what I tell my students at the start of the lecture: don't ask anything outside. But I think Roger has the answer to that. Roger.
I think it's very easy for people, especially the press, in the lay literature, and dare I say for regulators, to talk about PFASs as PFASs do this or PFASs do that, or they might do this or might not do that. And in fact, I believe PFOS and PFOA are different enough in their toxicological and health profiles to consider them as two separate chemicals. Yes, they do have overlapping toxicity and overlapping health associations, but as I said, I believe there is sufficient evidence to consider them as two separate entities.
Any other questions? I just thought I'd make a point that in the environment in Victoria, there's very little PFOA being measured. So we're certainly getting more of the PFOS and PFHXS, and not much of else either. So the two that we would be concerned about would be PFOS and the sulfonates. Yeah.
Excess sulfonate, yeah.
Just a question on behalf of some of the media that are watching that we put to you on their behalf. Given limited resources and what you know so far, how do you identify where we actually need to put our effort into where we need to test for higher concentrations, and where we are unable to actually identify exactly where it's coming from? How do we hope to control the future contamination if we don't know where it's coming from? Or yourself?
I think that's a question for you. Or for Roger.
Career limiting question there on my behalf.
Okay, well, I think many of you know that we've been doing a lot of work in Victoria in terms of understanding the spatial scale of common PFAS in areas that are not contaminated, because remember, we are working with areas that have got point sources, and that's quite different. So we've been trying to understand the concentrations they're not background, but areas around communities in soil, sediment and freshwater. And we've also been doing some work in biota. And so that is actually helping framing our assessment of exposure, because we are generally seeing that where we've got populations, large numbers of people and sectors, that we have increase in those PFAS. The other thing that's interesting though is you talk about drugs to try to eliminate more PFAS, but if wastewater treatment plants are a source of PFAS in the environment, we wouldn't want more PFAS in wastewater treatment plants, would we?
Oh, if we get them out of our bodies.
Yeah, but then they go into the effluent. Jochen? That's not, no. So anyway, okay, so clearly we have another question. Go, Roger.
I actually have a question that Jochen can answer. In your serum studies, Jochen, you've demonstrated a decrease in exposures and a decrease in concentrations. In those pathology samples, or in any of your samples, have you done any correlations between the clinical chemistry, total cholesterol, triglycerides, and the other information?
So in our pathology samples, we can't, it's all pooled. Because we rely on de-identified leftover pooled. So it's, that's bound by all kinds of ethics, you know. So we've done it in the firefight. We've done it in individuals. We do uric acid, cholesterol. We actually look at what other indicators. But we haven't found, but our numbers are far too small. So each dot on these serum samples was 100 people in one, or 100 samples in one pool.
Even though your samples are small, in those cohorts, have you seen any correlation with any of the clinical chemistry?
We don't do clinical chemistry in those pools.
But in your firefighters?
Okay. We have not seen the clinical chemistry going into the direction that the literature suggested. So I showed one little figure, and this is published, that shows that there was no increase with cholesterol, for example.
That's what we have found as well, but again, we've got very small numbers.
Yeah. I think firefighters are a particularly interesting group of people. Their actually body mass index is not a good indicator for their body shape. If you know what I mean. You know, they're really sporty.
Do you want to ask a question? Final question? No. Okay, we've got time for one question. Okay, any last takers before we finish this session? Okay. On that note, I'm going to stay there. I'm going to thank my colleague. I think you'll all agree that we certainly have had some really good information about what we know, right through from measuring the various PFAS exposure, potential for health effects, and some of the challenges that face us in the future. Please join me in thanking Professor Muller. Thank you. So we have we have another environmental science seminar series for May. Please have a look for when that comes up again, and I look forward to seeing you again. Thank you all for your attendance.